Hi there, it's Matt Walker here, and welcome back to the podcast, and welcome to a very special episode, one that I've been desperate to record for some time. This is about aging and Alzheimer's disease, and we will be weaving in commentary on sleep. I've already done a podcast previously on the interaction between sleep and Alzheimer's disease, and I'm sure we will come back to some of those features. But I have been working in the aging field since I was a much younger man, when I was starting my PhD, and it's continued throughout my career, but with a very strong sleep focus. Nevertheless, when it comes to the field of aging and Alzheimer's disease, I'm not an expert. And when you want to discuss this topic because of its seriousness, you reach out to an expert. One of the reasons that I ended up leaving Harvard Medical School and joining UC Berkeley was because of the gentleman I'm about to speak with, and that is Dr. William Jagust, or Bill Jagust, and you will hear me call him Bill because we're on good terms. Is that correct, Bill?
Oh, yes. Bill's fine, Matt.
Thank you very much. Bill, when you hear people speak about legends in their field, you kind of think, is that a little bit of sycophancy? But occasionally there are true legends in their field. Bill is that in the field of brain science. He is a neurologist and he does, all sorts of work. But one of his core research throughputs is aging and Alzheimer's disease. So, Bill, firstly, welcome to the podcast.
Thanks, Matt. It's great to be with you.
So maybe I've given a little bit of color, but could you perhaps maybe introduce yourself and briefly describe a little bit about your research focus?
Sure. As you noted, I was trained as a neurologist, and when I finished my clinical training, I was very fortunate to be in the right place at the right time. And I did what is essentially called a postdoctoral fellowship, where I went into a lab for 3 years and I learned about imaging. And at the time, frankly, I was really more interested in understanding how we could learn about the brain than I was interested in any specific problem. But my supervisor was studying Alzheimer's disease, and he kind of pushed me in that direction. And it wasn't very long before I just realized this was an incredibly important problem and that we were in the process of developing good tools to understand it. And that is essentially brain imaging. So ever since I was really a much younger man, I've been using brain imaging to understand how the brain changes as we get older. And I've been very fortunate to have lived through a remarkable expansion in medical and imaging technology to the point where we can now see things in the brain that we just couldn't even imagine seeing 30 years ago. So what I use mostly in my research is a technique called PET scanning, which allows us to inject radioactive materials into people, very, very low doses of radiation, so it's safe. And that allows us to see fundamental chemical changes in the brain that are occurring as people age. And we can relate those changes in the brain to changes in their cognitive ability, to their function, to their brain structure, to things like sleep and so forth. And a lot of the projects that we've done with you, Matt, of course, took advantage of those technologies.
When you're injecting those, what we call tracers, those radioactive tracers, in a way, is it that those tracers will then go up into the brain and they're almost sticky and they're selectively sticky? I'm just using sort of a metaphor, but they're sticky because they will latch on to specific either brain regions or they will latch onto specific pathological ingredients, things like beta amyloid that we've discussed on the podcast before and people may be familiar with. Is that what you're really wanting to do with those radioactive tracers? And do some of them do other things, like looking at sort of the metabolic activity? Help me better understand how those sort of radioactive tracers work in the brain and why they're so powerful that produce the sort of inspiration to do this work.
That's exactly right. You described it very well. I mean, what we do in this field is we design these molecules, basically, which are tracers, and we design them to have an affinity or to bind or to stick to a specific target. And that target could be a protein in the brain like beta amyloid. It could be a neurotransmitter receptor. It could reflect glucose metabolism or something of that sort. And the beauty of PET is that it's got incredible flexibility in terms of the kinds of molecules that we can design. So again, I've been very fortunate because during my career, molecules have come along that are labeled with radioactivity that bind to the amyloid, for example. Amyloid itself is actually fairly sticky. It sticks to itself and creates these deposits in the brain we call plaques. But it also allows us to stick this PET radio tracer onto it. And so we inject the tracer into a vein, and the tracer circulates around the body, gets into the brain, and then just sticks to the plaque. And so if we take a PET scan or an image, we can see where the amyloid is in the brain, but not only where it is, but how much of it there is. And we can bring people back in 6 months or a year or 2 years and see how it's changing. This has just been a phenomenal technology. Now, we've also studied glucose metabolism. As you say, we've actually looked at another abnormal protein in the brain called tau, which accumulates in pathological forms as another key hallmark of Alzheimer's. And again, we can label tau with this technique as well. What's beautiful about it is we get very accurate quantitation of what's happening in the brain, and the dose of radioactivity is so low that it's actually safe to repeat over time in people. So these are amazingly quantitative, sensitive, and very, very accurate tools for tracking these pathologies in the brain.
And in some ways, what I think, even back, gosh, when I was a younger lad at the Queen's Medical Centre in the UK, we often had to wait for patients who had different forms of dementia. We often had to wait for those patients to pass away and then do a post-mortem study and look inside of the brain to finally see if we could understand, where was the amyloid? How much amyloid was there? Where was the tau? How much of that tau protein was there? But now what you're telling me, Bill, which is part of this revolution, you can do that very early on, and you can do it as people are in their maybe 50s or 60s. You can already start to build up that almost time-lapse photography of how this accumulation cascade of Alzheimer's disease pathological ingredients is unfolding in a way that we never had the option because we never had these powerful tools. And PET has lots of flexibility. I mean, you've done amazing studies looking at other types of conditions, things such as dopamine within the brain, and obviously related to things like addiction. But what inspired you to specialize in the study of brain aging and Alzheimer's disease in particular? Was it in part this revolution of neuroimaging, or was there something else about brain aging in a healthy way and brain aging in an abnormal way, for example, Alzheimer's disease, that just as a younger researcher, you just thought, I just cannot get away. I'm just so fascinated. This is it for me.
I'll tell you one thing. It's interesting. I was not one of these folks who had a family history of Alzheimer's and was motivated for that reason. I talk to a lot of young scientists now, and it's really very rewarding to see people motivated by a desire to treat a disease, for example, that they've experienced firsthand from a parent or grandparent. But that wasn't me. I got into this at first really from a very technical perspective. And let me put it in the context of something that we call a biomarker. I don't know if your listeners are familiar with this idea, but this is something that's really gotten a lot of currency in the field of biomedicine and biomedical research. And a biomarker is simply a measurement that we can make that tracks either a disease or a process that's related to a disease. And they're really very broad. I'm talking about PET scans, but you can consider, for example, a blood test for your cholesterol to be a biomarker, or a blood pressure measurement can be a biomarker. So we know that if you go to the doctor and have a blood pressure measurement and it's high, we know that that's a sort of a biomarker for a risk for heart disease. So you'll be put on a blood pressure lowering medication, for example. So I was interested in this idea when it comes to brain aging. Some of the very first research I did was with a PET technology called glucose metabolism scanning. And that allowed us to look at the brain's metabolic profile. And way back in the 1980s, we began to see that there was a very characteristic pattern of abnormal glucose use that we saw in people who had Alzheimer's disease. And shortly after that, people began to describe this pattern in people who later developed Alzheimer's. So they were normal at the time, but then they developed Alzheimer's maybe 5 or 10 years later, and you could see this pattern in the glucose metabolism. We've subsequently learned that it's way more complicated than that glucose metabolism isn't really a great biomarker for Alzheimer's disease, but it was pretty good, and it was all we had at the time. I just started becoming entranced with this idea that we could understand what was going on inside the brain as people got older. And my interest just expanded from that relatively narrow technical enthusiasm at the beginning, wanting to understand if we could detect Alzheimer's disease before people had symptoms. Because with every medical condition, we know that if we can treat the disease before it's really serious, we have a better chance of curing it or at least having an impact. I also thought this might help us identify who's going to have an aging outcome that's really good. Which of us are going to live into our 80s and 90s and still be cognitively vibrant and intact, as opposed to those who are going to have cognitive problems. And then, of course, maybe there's something we can do to intervene to change the direction of these trajectories. So the whole idea of having a biomarker just got me at a very early stage of my career.
I love that word that you used, being entranced. And for people who maybe aren't scientists out there, when you are captured by what ultimately will be your field, it is such a beautiful description, an elegant description. It just comes at you and you become enthralled and entranced by this, and you have to follow it. It's just, it's almost like a compulsion. But coming back to it, before we speak about Alzheimer's, let me just start with brain aging. Help us understand what exactly is brain aging in the context of healthy brain aging, because I know that as we get older, our brains become less healthy over time. But people can progress into their 80s, 90s, and they have no clear signs of marked dementia. So they have progressed through aging in a healthy manner from the perspective of their brain. Help me understand, what is brain aging? What is healthy brain aging? And how does it evolve perhaps decade by decade? And this is obviously separate, as I said, from Alzheimer's disease, which perhaps we can come on to next.
Yeah, well, for the longest time, we had a lot of difficulty even knowing how to go about answering the question you just asked, because during life, you can't really tell what's happening in the brain. It's been a very slow progression of knowledge in this field. And to summarize where we're at, I would have to say that the changes associated with aging, when you take away everything that we want to call a disease, so Alzheimer's disease is one disease. Let's cast that aside for a minute. There's things like stroke or vascular disease that result from impaired blood supply to the brain. Let's put that aside. I could go on with lists of things that we should put aside, because it's very important to understand that as we get old, we're subject to more and more things that we call diseases. But not everyone gets these diseases, and many people live to be 70, 80, 90 without having brain disease. And what really has come out of this research is that what happens to your brain simply by virtue of growing old is remarkably minimal. It's not that much. We lose some brain connections. So these are called synapses, where one neuron will connect and talk to another. And I think it's pretty widely agreed that synapses are lost and connections between neurons are lost. But neurons or brain cells themselves, they don't really die in any substantial number. This loss of synapses is probably related to shrinkage of the brain. So that's something that happens as we get older. What I've just described is kind of the core of it. Now, there are changes in things like the connections between the brains, these long-range connections that we see on MRI scans, we call white matter. So there's changes in white matter. But there's really not amazingly profound changes in the brain other than really loss of synapses. And I guess I should also add, what happens in addition to this synaptic loss is that there are a few very small, specific regions of the brain where we actually do lose neurons. Most of the brain, we don't lose neurons. But in some parts of the brain, like in the parts of the brain that are responsible for dopamine, which degenerates when we have Parkinson's disease, we lose some dopamine cells as we get older. And we also lose some cells in a part of the brain called the hippocampus, which is very, very important in memory. And so there are small regions of the brain where neurons or brain cells are lost, but by and large, this is not profound throughout the cortex. And most of the neuronal loss that we see is very limited. Most of what happens in the brain is loss of these connections or synapses.
And what's interesting, I think, for the public to hear too, you would think, well, as my brain ages, people will have heard this. It's just lay street conversation. I'm losing my brain cells as I'm getting older. What you're really saying here is, no, you're not. But what this impresses upon all of us in neuroscience is that brain cells aren't necessarily the metric of the computational horsepower of your brain. They're necessary, but they're not sufficient. What is sufficient, in addition to the necessary, is having those brain cells connected to other brain cells. And the more interconnected those brain cells are, the greater that computational, that compute horsepower is of the brain. And as we get older, what you're telling us is that part of that natural aging where we just don't feel as facile as we did when we were in our 20s. And by the way, it's not just about how people think. It's also the brain controls how we move, how we do problem solving, how we make those new memories. All of these things require interconnected brain cells. And as we get older, interconnection is lost. One quick question. A lot of people listening may be thinking for either a relative or themselves, if they're in those more senior years, when should I be worried? When you see patients and you're speaking with them and they say, the other week, I was struggling to remember where I put my card keys, or I had my purse with me. And then I was thinking, where did I leave it in the house? Or Richard's friend who came over, I've met him a couple of times. I just can't remember his name. What are the signs when people should get nervous in terms of their memory signaling the potential of dementia? And when is it just part of healthy aging?
There's no simple answer to that. It can be very difficult when you're sitting with an individual and getting a history and even examining them to make that decision clearly. But I think there are some generally good rules. I mean, one of the things that's very common as we get older is forgetting nouns, especially proper nouns. You know, you're trying to come up with a word and you can't remember the word, or you can't, as you suggested, you can't come up with the person's name. Those are very common as we get older, and they're not particularly associated with Alzheimer's disease. Forgetting where you put things and left things, I think these are the kinds of problems that I think are fundamentally related to problems with paying attention. And this is another part of your brain, probably the frontal lobes, that are not really focused in on what you're trying to do. So you forget where you left something. What really is a clue to Alzheimer's disease is, I think, forgetting experiences that you've had, especially experiences that are important. I mean, if I asked you, you might not remember what you had for lunch 2 days ago. But if I asked you what you did this morning, or if I know that you went out to dinner or had a interesting event last night and you had no recollection of it, that would be cause for concern. So this is something we call episodic memory. And it's called episodic because it's based on an episode that happened to you in your life. And I think when you start to forget these things, that, you know, events in your life, especially ones that are particularly significant or salient, that's when I think the alarms should go off.
A supporter of today's podcast is Magic Spoon. I have had a love affair with cereal for most of my life, but when I started to sort of focus on wellness and health, I realized that most cereals, they were just really unhealthy, so I stopped eating them and life was much more miserable. And then my friend, Dr. Peter Attia, introduced me to a company called Magic Spoon. Magic Spoon is a fantastic cereal company. You can check them out. They have really healthy, high protein cereal. Magic Spoon, in addition to their cereals, they have now introduced Magic Spoon treats. And I have certainly added them into my panoply of things that I enjoy eating and that I use in terms of my fitness routines. They've got 11 grams of protein, just 1 gram of sugar, just 4 net carbs per bar. And they're just really well balanced if you're looking for this type of thing. There's tons of flavors to choose from: chocolate, peanut butter, blueberry, double chocolate, hashtag my favorite, strawberry milkshake, you name it, they've got it. Magic Spoon treats are available at your nearest grocery store. You can find them online. If you're looking for a healthy, fueling snack, I would suggest checking them out. For folks listening to this podcast, you can actually get $5 off your next order. Just go over to magicspoon.com/Matt Walker. And as I said, you will get $5 off there. Let's get back to the show now. Speaking about the alarms that go off, let's move into then Alzheimer's disease, which is really what we consider as non-normative aging. It's a disease, hence we call it Alzheimer's disease. We've spoken a little bit already about two of the, what we think are the pathological culprits in the brain that conspire well, actually, I'll leave that to you if they do conspire or not, but conspire together to produce this condition. Exactly what is Alzheimer's disease from the 30,000 foot perspective when someone says, yes, I know my mother has Alzheimer's disease, but we say that, but many of us don't really understand what it is and meaning what it is in the brain. Everyone will know the consequences of it. My mother is forgetful. She gets anxious. She gets worried. She perhaps has aspects of paranoia, but those are the symptoms. Underlying that brain perspective, what is Alzheimer's disease?
That's an important question. And I hate to backtrack and go from the 30,000 foot view, but let me start by saying, what is a disease? I think when we talk about what is a disease, we're talking about two things. One is a group of symptoms, and we call them signs and symptoms because signs are what the physician sees and symptoms are what the patient complains about. So they're symptoms, and they're associated with a specific abnormal process in the body. That can be things like cancer cells or blood clots or any number of things. So Alzheimer's disease is two things. It's symptoms of cognitive decline, and they're usually, if left unchecked, which they almost always are, progressing to something we call dementia. And dementia is a loss of functional and cognitive ability. So it's loss of memory, language, other kinds of behaviors that impairs the person's ability to function in their daily life. So that's cognitive decline and dementia are one half of the equation of what Alzheimer's disease is. But dementia has many causes, and Alzheimer's disease is a specific cause. It's associated with specific abnormal changes in the brain, which are essentially, as we've been discussing, the buildup of these two proteins called beta amyloid, we frequently call it amyloid for short, and tau. And when this amyloid and tau is deposited in the brain in sufficient amounts and in specific parts of the brain, and it's associated with dementia, then we say the person had Alzheimer's disease. Now, maybe we'll come back to this later in the talk. There's some discussion in our field right now as to how important the symptoms actually are. Can we say someone has Alzheimer's disease if they don't have the symptoms and they have the amyloid and the tau pathology? But that's a rather radical view, and not everyone agrees with it, but it is achieving more and more currency in our field.
So if someone were to say to you, what is our current understanding of what causes Alzheimer's disease? I suppose it's a tricky question. Causes would argue which is the finger that flicks the first domino cascade. And I don't mean it in that sense because I think we'll come on to risk factors later. But is it really about amyloid and tau? And some people listening may have said, well, hang on a second. I remember reading an article about that beta amyloid thing, and it seemed to be the leading candidate. And all of a sudden, there were news articles saying the amyloid theory of Alzheimer's disease is wrong. Where do we stand in terms of understanding these two pathological proteins? And how have you felt about that pushback regarding amyloid, either in the news or in the research field?
Yeah, I mean, you've hit upon really the central controversy in the field. And I definitely have feelings. Well, maybe I shouldn't say feelings. I have thoughts about it. I probably have feelings about it
let
too.
let loose on those feelings, Bill.
I'll try to keep it to thoughts. So let me tell you what I think was one of the most profound observations in this area. And that is, well, it's been known for a long time that sometimes Alzheimer's disease runs in families. And we kind of know that there's an increased risk if you've had it in a parent or sibling. But it's been well described that there are cases of Alzheimer's disease that are transmitted from generation to generation by a genetic abnormality. And these are very, very strongly inherited. Most people don't have this. It's a very uncommon situation. If you have this in your family, on average, a half of each generation gets Alzheimer's disease. That's on average, so it may not always be 50%. But this is what we call autosomal dominant genetic transmission. And this has been known for a long time. But in the 1990s, the genes that caused this autosomal dominant Alzheimer's disease were discovered. And what we have learned subsequently is all of these genes affect protein manufacturing mechanisms in the body that result in producing more of this beta amyloid. And people who have these gene mutations, whether you look at them with PET scans or whether you look at their brains on neuropathology, autopsy, they have Alzheimer's disease. They have amyloid plaques and these tau deposits we call neurofibrillary tangles. Basically, these genetic causes of Alzheimer's disease produce mutations that affect amyloid processing. And this is sufficient to cause Alzheimer's disease. So an abnormality of amyloid will cause Alzheimer's disease. So that's been tremendously revealing about the importance of amyloid in producing Alzheimer's. The other part of the story, though, is that when we've started to look at postmortem data, pathology, and more recently, PET data, one of the things we've learned is you can actually have quite a bit of amyloid in your brain. And now I'm not talking about the autosomal dominant genetic cases. I'm talking about just studies of older people. We bring them into the lab. Many laboratories have done this and found that there are plenty of people in their 70s or even 80s who have a lot of amyloid in the brain, but they're cognitively normal. They don't have dementia. And so that was one of the first chinks in this wall of amyloid. How can amyloid cause Alzheimer's disease if you can really have quite a bit of it in your brain and not suffer from any problem? Well, it turns out that you have to have this tau in your brain to cause cognitive decline. And we've now seen that you never have high amounts of tau in the brain unless you have amyloid. And in fact, if you have amyloid in the brain, and now we can follow people over time with PET scans, if you have amyloid in the brain, you're going to be depositing tau in the brain at a faster rate than if you don't have amyloid in the brain. So the story that I think is really the most convincing is that this amyloid comes along and does something to some tau molecules that we have in the brain. And actually, we all have some of this abnormal tau in our brain by the time we're 70, but we don't all have amyloid. And when the amyloid comes along, it interacts with this tau in some way that we don't understand and causes the tau to accumulate more and more and more. And that spreads throughout the brain and causes the cognitive decline. So the argument about what causes Alzheimer's disease, it's kind of a semantic argument. You can't have Alzheimer's disease without lots of tau in your brain, but you're probably not going to have lots of tau in your brain without a lot of amyloid in your brain. And so I think if you get this discussion, you'll understand why some of the controversy has been around, which is to say that, well, you can have a lot of amyloid and not have Alzheimer's disease, but it's not simply the amyloid that's causing the problem. And so I think we have a pretty good model of what Alzheimer's disease is based on these kinds of studies that we've now been doing longitudinally that really show us that the amyloid drives this accumulation of tau and that in turn is what drives the problems with cognition.
And so amyloid is necessary again, but not necessarily sufficient, because you can have lots of it in the brain and not have Alzheimer's disease. However, what you can't have is a lot of this next step piece of pathology, and I'll explain why I say next step, but the tau protein, you can't have lots of that if you don't already have a quite considerable burden of amyloid. And therefore, amyloid seems to be almost a pump priming, in a bad way, development that then can subsequently lead to the accumulation of this other protein related to Alzheimer's called tau protein. And then that tau protein, and you tell me if this is the right conceptualization, ultimately, together with the amyloid, they are both the arbiters of the brain cell damage and perhaps the loss of synapses, the loss of the actual brain cells themselves. And it's that loss of brain cells and synapses that ultimately produces the symptoms of Alzheimer's disease. Is that the right way to think about the cascade
Yeah,
of the domino effect?
absolutely. I mean, I think there's strong evidence that both amyloid and tau have very harmful effects on the brain cells and the synapses. Tau is probably more problematic. Yeah, that's exactly the way it works, that these abnormal proteins affect synapses, they affect cell-cell communication, they also affect health of the brain cells, and these cells seem to die. Very different than normal aging, when the cells really don't die. This is what ultimately leads to shrinkage of the brain, and you get really massive shrinkage of the brain in Alzheimer's disease, and very obviously substantial loss of cognitive ability.
So let me try to then loop back to things that we spoke about at the start, which were the powerful tools of PET imaging and these different tracers. At this point, we've mapped out what Alzheimer's disease is, and we've understood part of the mechanism, this cascade of these different pathological ingredients. But it turns out, for people listening, that Alzheimer's disease pathology, amyloid and tau, don't necessarily accumulate all over the brain in an even distribution, like butter spread very evenly over a piece of toast. Instead, they seem to build up in some parts of the brain, but not other parts of the brain. And one of the reasons that we know this is not just from those post-mortem studies that used to unfold, but because of these powerful new brain imaging tools, where within a living individual, you can actually see where this amyloid and where this tau is building up. Where are these pathological ingredients building up in the brain of a patient who is progressing into and then full-blown Alzheimer's disease? And what does that tell us about explaining the symptoms that they will experience? But also, what does it tell us, if anything, about the nature of this disease, both regions that are impacted, and maybe what does it tell us about the parts of the brain that aren't impacted? So really several questions there, but where do they build up? How does that explain the symptomatology? And maybe does it tell us anything profound about Alzheimer's?
Yeah, again, you've hit upon a really a central question, actually, not just in Alzheimer's disease, but pretty much in all neurological diseases. And that is this question that we in the neuroscience world refer to it as selective vulnerability. That is, there are areas of the brain that are selectively vulnerable to the pathology of Alzheimer's disease. And they're different for amyloid and tau. The amyloid gets deposited in actually fairly widespread regions of the brain, and they're regions of the brain that are largely associated with very complex cognitive function. Now, again, your listeners who haven't really thought a lot about how the brain is organized, you may immediately think, well, the brain is very complicated and does very complicated things. And for sure, even the simplest thing that the brain does is very complicated. But amyloid doesn't tend to get deposited, for example, in the parts of our brain that deal with sensation or motion. So the parts of the brain that are responsible for moving your arms and legs, this we call the motor cortex, and part of your brain that's responsible for sensation, the sensory cortex. Amyloid doesn't really get there. It doesn't get in the coordination centers of the brain, like the basal ganglia or the cerebellum. It really is deposited in regions that we call association cortex. And these are fairly large areas of the brain that are dealing with very complex cognitive functions like judgment, planning, recognizing things in your environment, memory, things of this sort. Why does it deposit in these brain regions? Well, we don't know. That's the simple answer. But I think the leading theory is that these brain regions are all very, very busy. They're very, very highly active metabolically. They're very connected to one another. And it may be that there's something about the need for this high level of activity and metabolism that's driving somehow the deposit of amyloid, although we really don't know this. So that's amyloid.
What you were saying there in some ways is regions of the brain across our lifetime that seem to undergo the greatest activity demand, almost like a car engine. There are some parts that just have high amounts of wear and tear. And it's those regions that seem to be especially vulnerable to the building up or the depositing of amyloid within the brain.
Yeah, exactly. And, you could say these are the brain regions that never really turn off. I mean, I don't know if our brain ever turns off, but there are certainly brain regions that are, as you say, highly active, strong demands on them much of the time. So the tau is a little different. And the tau brain regions overlap to some extent with the amyloid regions, but not entirely. And the tau regions tend to occur in a part of the brain we call the temporal lobes. The temporal lobes are really interesting because they're very strongly associated with memory and memory ability. I don't want to overstate the case because the tau, it can spread in later stages out of the temporal lobes to other parts of the brain. There's very, very good evidence that the temporal lobes are the early place the tau deposits, and it's really remarkably vulnerable.
The temporal lobes that built, those are the things that sort of sit essentially not too far away from your ears. Temporal sort of the far left and the far right flanking slabs of the brain rather than the back of the brain or the middle or the front of the brain.
Yeah, that's a good way to describe it. Exactly. They're kind of like almost like little flaps that surround the lateral surfaces of the brain. The interesting thing about these brain regions in the temporal lobe is that they're very functionally important in memory and in linking up memory to other things like perception. So how do we know that baseball is a baseball? Well, we visually perceive it, but then we have to connect it to something in our memory to remind us that it's a baseball, or maybe even the baseball that you had when you were a kid, so you can remember a lot about that object, but you need to hook up this perception to the memory to really decode what's happening in your environment. And we think the reason that tau is deposited in the temporal lobe has to do with the patterns of connections between neurons. And there's now tremendous evidence that tau spreads through neurons and through connections between neurons. And I can't really tell you why it starts in the temporal lobe. In almost all of us, it starts in a tiny little part of the temporal lobe called the entorhinal cortex, which is part of the memory system of the brain. So we almost all have some tau in that part of the brain by the time we're in our late 60s or 70s. In most people, that tau just sits there and doesn't seem to cause much trouble. But when the amyloid comes along, the tau spreads out of this tiny brain region throughout the temporal lobe. And there's really a lot of evidence that it spreads through brain cells, through neuron connections. And the reason then the tau is in the temporal lobes is because the patterns of connectivity of that brain region where the tau initially deposits. Now, why it initially deposits there, it's not really clear. But once it's there and starts to spread, it probably shows up where it does because of the way cells in the brain are connected to one another.
What's interesting for folks, and I mentioned this on a previous podcast, is the work that you and I have done, Bill, and others have done great work in this. The amyloid that builds up in the brain doesn't necessarily, again, build up all over the brain. It builds up in these highly metabolically active regions. Unfortunately, what we also know is that the parts of the brain that are attacked by amyloid are the very same regions that help generate the deep non-REM sleep that we experience every night. And the unfortunate part of that is it's the deep non-REM sleep that we have each night that provides almost a cleansing mechanism to wash away some of the metabolic detritus that builds up when we're awake. And that includes, based on the science that is out there right now, beta amyloid and also aspects of tau protein. So in other words, it's almost this vicious cycle, and it's a very unfair cycle, which is as amyloid builds up across the lifespan, it builds up in the parts of the brain that generate that deep sleep. And the deep sleep is normally there in part to do lots of things, but one of them seems to be cleansing the brain of amyloid and tau. And so the less that you're having, we believe, the more that may be building up. The more that builds up, the less deep sleep that you have. The less deep sleep that you have, the more that builds up, and so on and so forth. And it's almost this escalating, almost like compounding interest on a loan. And in some ways, that's what we see in Alzheimer's disease with amyloid. It's not necessarily, and you tell me, well, a direct linear 45-degree line that climbs up. It starts off and then it escalates almost exponentially. And then at some point, it just, reaches its maximum. A sponsor of today's show is Inside Tracker, which is a service that comes out to your home, and they will analyze your blood and your DNA to know precisely what is going on inside of you. Hence the name Inside Tracker. They look at your blood, your metabolic signals, your hormonal health metrics, and then they give you a personalized, actionable set of lifestyle changes in response to that readout. And the goal there is to improve your health. I was looking and informed they have some new cardiovascular and new hormonal biomarkers that I'm particularly interested in. One that I'm focused on is something called ApoB, which is an absolutely critical heart health measure. And I get it done now with them somewhere between 4 to 6 times a year. Why? Well, my family unfortunately has a strong history of cardiovascular disease, so I am checking that pretty ruthlessly. And by the way, I do buy the product myself out of pocket. I don't want to fall prey to any of those trappings and undue incentives. Although, with full admission, I still use my own discount code that you can use to get some money off. And that code for you is insidetracker.com/Matt Walker. So just go over to insidetracker.com/Matt Walker. And again, if you want to get that discount, it is insidetracker.com/Matt Walker. Thanks very much. So that's a little bit about the sleep component. But what you're saying there too, which is, I think, a new vision regarding tau that's come along, you tell me, in the past 5 or 8 years, you've done some seminal work here. Tau doesn't just seem to appear in parts of the brain. It almost seems to move through the brain, that it has this trajectory, that it almost uses the connected network of the brain in these memory centers in the far left and the far right lobes of your brain. And it's almost burrowing its way through the brain. Is that what you're telling us, that tau seems to move along the neural connections within the brain and it sort of transitions and throughout the brain in that way?
That's exactly right. We know from studies in animals that tau can spread from neuron A to neuron B, just like traveling down a road. And when it gets to the synapse, the synapse is a little space between neuron A and neuron B. There's very good evidence that the tau just sort of swims across that synapse and ends up in the next downstream neuron and keeps going. Part of what's happening is as it comes in contact with other tau molecules, it transforms them probably and makes them abnormal. So one of the processes that's underlying all this is another, I'm getting fairly into the weeds here, but is another process called misfolding, what happens is if you have an abnormal shaped protein, and it could be amyloid or tau, that comes into contact with a normal shaped protein, it induces the normal protein to fold in a way that's abnormal and pathological. And amyloid does this to other amyloid molecules, and tau does this to other tau molecules. And there's even some question as to whether this can happen cross molecule, but that's not really known.
It's almost like a game of tag. And I remember back, as I said, when I was at the Queen's Medical Centre in the UK, it was right in the era of what we call mad cow disease, or Creutzfeldt-Jakob disease is the more medical term. And that was the first notion that we had, that this misfolding pathological change in the brain could almost act like a virus, that proteins could almost be a virus. It's like a game of tag, that this bad form of the protein, this misfolded form, races along and sees a good form of the protein, and it says, tag, you're it. And that previously healthy protein now becomes misfolded and it goes off. And so you get this almost viral transmission. And what you're saying is that in Alzheimer's disease, one of our emerging theories is that amyloid and tau can both also act in a similar way that these prion proteins did in mad cow disease. And they can start to tag other good proteins, misfold them, and hence you get this cascade of growing Alzheimer's disease in the brain. That's fascinating.
I don't think there's much doubt there's always some doubt, I guess. There's really very strong evidence that this is exactly what's going on. And it's part of the reason that things can get worse relatively quickly, because the more of these abnormal molecules you have around, the faster they induce other molecules to become abnormal. So this misfolding is really a core feature of the problem. But yeah, we're hitting on some very fundamental concepts in biology. This prion protein was discovered by a neuroscientist named Stanley Prusiner, who won the Nobel Prize for it. So it's a really profound observation about infection and disease.
So let me move us past some of the mechanism and return to a slightly higher level. In your experience, vast as it is, what would you say are the major challenges in diagnosing Alzheimer's disease at an early stage? Because people listening are either going to have family members who are in the grips of Alzheimer's disease, or they are concerned about them, or there are people listening who start to have this sense that I'm just not operating in the way that I used to, and I'm getting up there, I'm retired now. What are the challenges that you see in diagnosing Alzheimer's disease quite early on? As you mentioned, the earlier we diagnose something, typically the better in medicine. Tell me about that.
I want to rephrase your question, if you don't mind.
Every time you rephrase anything of mine, I'm always thinking, I'm going to steal that, because that's exactly how I've said. Rephrase away, Bill.
Okay, so I just want to put this in the perspective of clinical medicine, because your question is completely fair. But I think in the context of clinical medicine and patient care, it always comes down to, what can we do for someone? So I can talk for a few minutes about how we might be able to detect Alzheimer's disease early. But the problem is, right now, we can't do anything about it. We're just at the stage of getting what may be effective therapies. So what I want to do is tell your listeners what we can do for people who have Alzheimer's disease and where I think that field is going, and then come to the point of who we want to detect early and how. I think in the last couple of years, we've really seen a remarkable series of clinical trials that have used drugs that lower brain amyloid. These are drugs that are, at the current time, they have to be infused intravenously, although companies are developing other modalities of administering these drugs. But right now, with these infusions, what these drugs do is they're antibodies, and they seem to take the amyloid out of the brain. And how do we know this? Because we can do PET scans. We can do PET scans before we administer the drugs and then after we administer the drugs, and we can see that the amyloid can be really massively cleared from the brain. Not usually entirely from the brain, but sometimes entirely from the brain. Usually we can take out a whole lot of amyloid. So these drugs are very effective at removing amyloid. Now, one of the reasons that that the amyloid idea has been sort of poo-pooed is that we've had drugs like this for about 10 years and they haven't really worked. They've pulled amyloid out of the brain, but they haven't made people better. But in the last year and a half or 2 years, we've had a series of drug trials that have removed amyloid from the brain and slowed the rate of progression of the disease. And the reason that these drugs probably work better than previous drugs is because they were just way more effective at removing amyloid. So they really just suck out a lot of this amyloid from the brain. These are people who are having symptoms, so they're already sick. But if you follow them over time, you see that the people put on these drugs decline at a slower rate. This is a real difference in the way we think about Alzheimer's disease, because this is a drug that's targeted on what we think is the fundamental problem in the disease. And we think that if we remove amyloid, we can slow the progression of the disease. Now, the question is, can we do this in people before they have symptoms? And the answer is we don't know, but that's the hope.
The reason that you would want to do that is because, going back to the patient who had Alzheimer's disease, you just told us that when we use these new drugs to pull out that amyloid, the rate and the speed at which they are going to decline in terms of their Alzheimer's disease is slowed dramatically. And some people may say, well, hang on a second. Why don't those drugs then put my father or my mother back to normal? Why don't they actually return them back to the normal state? And in some ways, is the argument there, firstly, they've already suffered a lot of brain cell loss and brain cell damage because of that amyloid. So when you pull it out, yes, you progress now at a much slower rate, but you can't get back that which you've lost. And therefore, it leads to your second point here, which is, if that's the case, yes, we would still want to do that, perhaps ethically, to slow that rate of decline down in those who already have Alzheimer's disease. But the much more powerful benefit of these drugs is not getting to that late stage, but intervening at the very earliest stage when we see just the earliest signs of this. Is that the right way of helping people understand why those drugs aren't going to put their mother or father back to normal, and why prevention, as you're moving on to it here, is that much more important?
Yeah, that's true. And I think the key to understanding is the brain really has minimal to no capacity to repair itself. So once the damage has been done and the neurons have been lost and the synapses have been destroyed, the brain can't repair itself. And so the hope that this would improve people, it's just not reasonable. And I think with all diseases, even ones with organs or tissues that are more amenable to repair, we know that the earlier we intervene, the better the outcome. So I don't want to suggest that it's pointless to intervene at later stages. I mean, many of the people in these trials had actually very mild Alzheimer's symptoms. And if you can keep someone in a stage where they may be experiencing significant memory loss, but they still can go about a lot of their daily activities, you've done a lot of good for them. But I think clearly the better the person starts out, the more likely they're going to remain functional and able to enjoy life. So, yeah, I mean, I think we're at a stage now where there are a number of trials underway where people who are cognitively normal are being recruited to participate in these studies. Now, we couldn't even think of doing that 10 years ago because part of the story here is these drugs are not entirely benign. They have some harmful side effects, and you have to balance the side effects with the benefits. And you really don't want to give a drug that has potential side effects to someone who doesn't have Alzheimer's disease, right? I mean, that would be terrible.
Right.
So 10 years ago, we didn't know who had Alzheimer's disease. So now we do. If we want to decide that Alzheimer's disease is the presence of these amyloid plaques, then maybe we can just treat people who have amyloid plaques and not worry about whether or not they have symptoms. And this comes back to something I said very earlier in the podcast, where I mentioned that there's this somewhat radical notion that if you have the amyloid in your brain and you don't have symptoms, you still have Alzheimer's disease. And it's very controversial and it's a little bit tough, right? Because there are some people who have this amyloid in the brain who are destined to have problems maybe in the next 5 years, maybe in the next 10 years. But there's other people who might not have problems for 20 or 30 years. This is really one of the big questions. Can we identify people who have amyloid in the brain And can we identify people who have amyloid in the brain who are at very high risk of getting sick or having symptoms? And the short answer is we can definitely identify people who have amyloid in the brain. And we can do it with a PET scan. That's very expensive. But more and more, there are now blood tests to detect amyloid. And we can do a blood test and see if a person has amyloid. And actually, we can put someone on an amyloid lowering drug based on a blood test.
I mean, that's remarkable because it comes right back to what you were saying early on in the podcast regarding diagnostic tools and intervention. You said, well, if we look at someone who has hypertension, high blood pressure, we have a biomarker for that. If you look at someone who has high amounts of cholesterol in the bloodstream, then we can put them on a medication. Why? Because we have a biomarker for that. This is why this revolution of the biomarkers for Alzheimer's disease, both within the brain, PET scans, but now circulating in the bloodstream, are so important because even at the time of developing those PET scans many decades ago, there was no hope of putting someone on an Alzheimer's disease drug for prevention. But we knew that that potential could unfold in the future, and therefore getting that biomarker in place was going to be an essential prerequisite. And this is why sometimes blue sky research is so critical. Yes, there's nothing we can do about it right now. And some people would say then, so why develop a biomarker if you can't do anything about it? Right now, we may not be able to, but give me 10 years or give the field 10 years, and we could get there. A long-standing partner of today's podcast is Element, spelled L-M-N-T. Element is a sugar-free electrolyte drink mix. And for me, it was the sugar-free part of that made me start using it. Gosh, probably 2 years ago now. And here again, by the way, I do buy my own supply just to stay objective. But beyond having the electrolytes in the right balance for human physiology, the other thing that I'm a fan of regarding Element is that it's scientifically backed. If you look up the people who created the company, you'll find some pretty heavy-hitting physiology, biochemistry, pharmacology knowledge. All of that's baked into the product. And again, it all comes without the downside of any sugar. And now, since I am such a devotee of their product, and P.S., I really do use them each day. I use it for my workout, and then I use it in my protein shakes. I really do, folks. I really do use it. I badgered them, and they have agreed to give you fine folks an 8-count completely free sample pack with any purchase of Element. And to get that, you do have to use the following link, and that is lmnt.com/Matt Walker. So if you'd like some free product with your purchase, just go to lmnt.com/Matt Walker, and you can pick up your free swag. So I think what's nice, Bill, so far, you've helped us understand a little bit about what brain aging is, what Alzheimer's disease is, the underlying mechanisms, how you think about the interaction between these pathological ingredients. We've spoken about the clinical trials, developing drugs that can at least pull out beta amyloid in the brain, and now how we can map your disease risk based on these brain scans and more recently on your blood biomarkers. I suppose at this point, people are starting to think, that's great news, but right now, at this moment in my life, depending on what age you are, no matter what the age, could you discuss any correlations as we move into this question of to de-risk my chances of developing Alzheimer's disease? In some ways, I'm asking a question, what are the correlations between lifestyle factors and brain health in general? But then also, what are the risk factors for Alzheimer's disease specifically? And how can I think about that as a man who now is in these sort of, as I mentioned, foothills of middle age. Tell me, what are the correlations for brain aging, correlations between lifestyle factors, risk factors I should be concerned about? Tell me, tell me.
I think it's a fair question for lots of reasons, not the least of which is intervening medically in a disease is fine, but it's really preferable to reduce the incidence and prevalence of the disease if you can, because you can do that much more cheaply and much more broadly. I always argue from the perspective of cardiovascular disease because we understand it really well, having tons of research for 60, 70 years. And probably the single biggest thing that affected the prevalence of heart attacks is smoking cessation. People just smoke much less frequently than they did 50 years ago, and that's had a profound impact on heart disease. So if you can intervene in lifestyle, the chances are pretty good you can have a big impact if the lifestyle really is on the pathway to the disease. So what is it with Alzheimer's? Well, I think if you look at the data in the aggregate, there's a tremendous amount of evidence suggesting that healthy lifestyle lowers your risk of Alzheimer's. And what do I mean by healthy lifestyle? So these are things that the things I'm about to tick off are things that I'm reasonably convinced of because they've been in more than one study. One of them is physical exercise. It seems pretty likely that people who participate in various levels of exercise, it doesn't have to be running a marathon, but even walking has been suggested to reduce your risk of Alzheimer's. Diet, so-called heart healthy diet or Mediterranean diet that's high in vegetables, legumes, low in saturated fats, low in meat, and higher in omega-3s and fish. Those seem to reduce your risk of Alzheimer's disease. Staying cognitively active, that's not simply a matter of doing puzzles and games, but it's staying engaged with life, whether that be social or doing things like going out with friends and interacting socially or reading or things like that. People who do those things seem to have a lower risk of Alzheimer's disease. Sleep, I'm sure you know very well. I'm sure you know very well. There's a tremendous amount of data to suggest that people who report disruption in sleep versus those who report restful and longer sleep seem to have a lower risk of Alzheimer's disease. That's reporting, but in addition to that, physical measurements of sleep, whether it be the kinds of things you do with polysomnography and actigraphy, things like that, seem to be associated with lower risks of Alzheimer's disease. Oh, and then I think the other thing that isn't quite maybe falling in the category of lifestyle, but keeping up medical care for other kinds of things, especially vascular disease. So people who have hypertension, diabetes, and high cholesterol, treating those cardiovascular risk factors, as we call them, seems to also lower the risk of Alzheimer's disease. So these are all very strongly associated with Alzheimer's risk. But there's one caveat that we have to be very clear about, and that is that these are association studies. So they show that there's an association, but for the most part, they don't really prove that these lifestyle factors cause the outcomes we're talking about. So in order to do that, we need to have a randomized clinical trial where we put people in one arm or another arm of a trial and treat them with a lifestyle intervention. And there are now been a few of those, and they look promising. There's a big one going on in the United States now called the Pointer Study. We don't have the results from that. But there's one that was done in Finland a few years ago called the Finger Study. And that showed a benefit to what we call these multi-domain lifestyle interventions, where people are taken and randomly assigned to different kinds of interventions. And there have been some benefits in those studies as well. I think it's not clear that these are really causal. On the other hand, I don't know who's going to argue with me that it's not good to do exercise and get a good night's sleep.
Certainly not me. In some ways, it's a small price to pay because those things typically don't have many downsides to them. We were speaking about the idea of these new Alzheimer's disease drugs, and you were saying, well, they're not without their side effects, so you have to be very thoughtful. But eating in a healthy manner, exercising and sleeping sufficient amounts with good quality and quantity, these things typically have a very minimal risk profile. So in other words, What's to lose by at least engaging in them, even if the science right now doesn't demonstrate with astounding reassurance that they are causally, non-negotiably, the things that are going to keep you away from the door of Alzheimer's disease? To me, it's well worth rolling the dice and betting on those things. We've covered many of the questions I was going to ask you, but one thing I wanted to ask about in terms of risk factors are genetics. I know that we spoke about genetics just early on in this episode, but people may be familiar with these kits that you can buy online, things like 23andMe, and often in there is a description of your risk for Alzheimer's disease based on a particular gene. And I just did an AMA episode a while back on this podcast, and I spoke at length about the different, what we call the ApoE combinations. But just for folks who've not listened to that, tell me a little bit about how you think of what's called the ApoE gene and its contribution to Alzheimer's disease as a risk factor.
Yeah. I want to make sure first that we distinguish this from what I talked about earlier, which were these gene mutations that cause what we call autosomal dominant Alzheimer's. Those families of genes are very, very strong causes of Alzheimer's, and they're extremely rare. But ApoE is very different. It's much more common, but it's much weaker. So let me explain the genetics. It's pretty simple. This is a gene that comes in three forms. They're not named very creatively. They're just 2, 3, and 4. They come in form 2, form 3, or form 4. And everyone has 2 copies of this gene. You get one from your mother and one from your father. So there are various combinations you can have. You can have 2, 2, 2, 3, 2, 4, 3, 3, 3, 4, or 4, 4, right? So there's 6 different combinations. So it turns out that if you have a copy of the 4 allele, which is not super common, but about 15% of the population has one. If you have one copy of this E4, your risk of Alzheimer's disease is increased. And it's a little hard to say exactly how much because it may differ in different racial or ethnic groups and may differ by sex, but it's probably on average about 3-fold the risk if you don't have an E4. And if you have 2 copies of this E4, your risk is even higher. It may be even 10 times higher than someone who has no copies of the E4. So it's a risk allele. It's not a cause. What does that mean? That means you have a higher chance of getting Alzheimer's, but it doesn't mean you will get Alzheimer's. And if you look at people who have Alzheimer's, about 60% of them have an E4 allele. But that leaves a lot of people who don't have one. So you can get Alzheimer's without having this E4. And you can also have the E4 and not get Alzheimer's, or maybe you're destined to get it when you're 90, which means you may never live to get it. So the E4 allele is a risk factor. It is very, very well replicated in the scientific literature. There's really no doubt about it, but it doesn't really cause Alzheimer's. It just increases your risk.
So now moving past this notion of risk factors and lifestyle changes that we can make to de-risk Alzheimer's disease, looking ahead, and I know it's always dangerous in research to do future forecasting, but how do you see the field of Alzheimer's disease research evolving in, at least in the next decade or so?
Well, let me preface this by saying that I'm an optimist.
I'm a rational optimist, I would say, but I think you are too.
Yeah. So I'm tending to look at the bright side. You know, all kinds of things can derail the idea that I'm about to suggest. Here's what I think is going to happen. I think we'll get drugs that lower amyloid, and we may have drugs that do other things as well. The amyloid lowering drugs are they're really going pretty fast and furious. There may be drugs that lower tau, and there may be drugs that do things like affect the way the amyloid folds, and so prevent the folding that is, we think, pathological. But at the very least, we have the amyloid drugs. And I think they're getting better, and they're also getting to a point where we may be able to administer them through, an injection in the muscle rather than an intravenous injection. And who knows, maybe someday there'll be something that you can take orally, but we're not there yet. So I think these drugs are getting better, both in terms of how well they lower amyloid and the ease of administration. And there may be other drugs that come along. And I think there are clinical trials going on to tell how early we can detect Alzheimer's. So I think what could happen is that when you get to be a certain age, could be in your 50s, more likely maybe your 60s, you'll get a blood test at your doctor's office, kind of the way men get prostate blood tests now. And it'll show your amyloid level. And maybe if it's above a certain level, you'll have a PET scan to confirm it and to sort of tell how serious it is. And then, depending on the results of those tests, you'll go on a drug and it will prevent you from getting Alzheimer's disease.
It's a remarkable vision to paint, but I agree. I don't think it's necessarily one that is science fiction. It's certainly science prediction, and it could be science fact in the not too distant future. What I've not done yet, which is so unfair to you as well, Bill, I've not really delved into your research specifically, but rest assured, listeners, a lot of the things that we've discussed so far, Bill has made or has been the fundamental contributor to, and he'll reject that because he's modest. But coming back to it, what would you say is the most surprising finding you've encountered in your own research in this area?
Well, I'll tell you, I think there are a couple of things that really surprised me. One is how much amyloid you could have in your brain and still be cognitively normal. When we did our first amyloid PET scans, we were surprised to see how much amyloid there was in the brains of our research participants who were doing extremely well. People living very active lives, nevertheless, had quite a lot of amyloid in the brain. So that was kind of surprising. The other thing that was really quite interesting was the pattern of tau. And there were two things about the pattern of tau that were really kind of remarkable to us. One of them was, I don't know if you remember back to what I said earlier, that at the beginning of my career, we had seen these glucose metabolism patterns that we felt were reasonably good at picking up who might have Alzheimer's disease, that these changes in the brain and glucose metabolism was predictive of Alzheimer's. Well, when we started doing tau PET scans, we were also doing glucose metabolism scans at the very beginning. And we found that the tau PET scans matched up almost perfectly with the glucose metabolism scans. So what was really kind of amazing to me was that I had started in this field, when I was a pretty young man. And maybe 25 years later, we did experiments that explained what we discovered at the beginning of my career. I didn't understand why we were seeing those things at the beginning of my career, but toward the end of my career, I understood the reason we saw them was because they had been caused by the tau in the brain. We spent a lot of time when I was younger trying to understand what caused those changes. We actually had studies where we looked at postmortem pathology and tried to relate it to the scans, and we could never really quite get it straight. But I think it's very, very clear that these patterns in metabolism are explained by tau. And then I guess I would just say one other thing, which is that we started to look at the systems in the brain that are responsible for memory. That was probably, only about 5 years ago that we were mapping out these brain networks involved in memory. And a very talented graduate student in the lab was able to do some experiments that showed really quite clearly that these memory networks explained where the tau was in the brain. And really, it was this story that I was telling you about tau spreading from one brain cell to another really explains sort of how tau gets where it is. But seeing this in our data and seeing that once we defined these memory networks, it explained where the tau ended up in the brain. That was a really, maybe surprising isn't the right word, because that was our hypothesis, but it was very exciting to see it.
And it's almost then it sounds as though tau is fiendishly selective in its appetite of geographical destruction within the brain. In other words, it's almost, and again, this is not the way to think about it, I'm just sort of anthropomorphizing, but it's almost as though tau knows where the memory networks are within the brain, and it seems to have this blinkered exclusivity to it, and it just goes after those memory networks. It's not as though, by the way, for folks listening, what Bill just mentioned there, it's not as though there is a single part of the brain that is packaging all of our memories together exclusively. There are some parts of the brain that if you damage them, you can't form or hold on to new memories. But there is an entire network, there's an entire set of scaffolding throughout your brain that is very specific to memory function. And what you're telling us based on your research is that it was very clear that we had this distribution of tau, this pattern of tau in the brain, and it was kind of strange. Why is there this pattern? Why is this very specific signature? And all of a sudden, when you figure out the network in the brain that is the memory network, it's almost a carbon copy for the tau distribution signature profile within the brain, and suggesting that tau, it seems very selectively, conspires to go after exclusively the memory systems of the brain. Is that really a fair sort of assessment?
I think it's true at first. Eventually, tau gets out of that system and causes a lot more damage, but it's certainly the beginning of the story. It's absolutely correct.
So, wrapping things up, what message do you want to leave people who are listening here regarding brain aging in general, and then separately regarding Alzheimer's disease specifically?
Well, I think on a practical level, a good message is that brain aging in and of itself, it doesn't seem to necessarily cause incapacity. And that if you or someone that you care about is experiencing significant decline in their abilities that impairs their ability to function, that's not just getting old. That's a disease. It's pretty good chance that it's Alzheimer's, but not necessarily. It could be something else. We talked about diagnosis in the context of early treatment for Alzheimer's disease, but there are a whole bunch of other things that can cause similar symptoms. And some of them are things like infections or brain tumors that can be treated. So I think probably a really crucial and not very complicated, but extremely practical message is that in older age, the significant loss of cognition and function isn't just getting older, it's a disease. And I think it is really important for people with those kinds of symptoms to get medical attention.
Thank you, Bill, for both giving people that message, but also just the contribution that you have made to the research field. You probably don't know it, but you've had a huge influence on how I think about aging and Alzheimer's disease, and the inspiration driven from you to do this type of work. But for what you do for the world in terms of your impact on understanding how the brain gets older, but also how it goes through this really despicable, vicious condition called Alzheimer's disease, I want to thank you for that. I know you're desperately modest and you will reject all of that, but say nothing and just accept that the people listening, I'm sure, have immense gratitude. Is there anything else that you would like to leave us with? Do you have any social media accounts that people can follow you on? Where can people find you? How best can they understand? There's lots of great talks, by the way, on YouTube and other locations by Bill, and talks, both scientific talks, but also general public talks that will really help you understand Alzheimer's disease. But where can people find you, Bill, as we sign off?
I avoid social media assiduously.
That's why you're so mentally well adjusted relative to me.
I do have a lab website, and it's very simple. It's Jagust Lab. If you just type Jagust Lab, that'll probably get you there. And it's
Jagust. That is Jagust.
Yeah.
Jagust.
Jagust, LAB, one word, and it's .neuro.berkeley.edu. But I think if you just type Jagust Lab in Google, it'll get you there pretty quickly. And yeah, you can learn more about what we do in the lab. You can find contacts and things if you're interested. And yeah, thank you so much for your kind words, Matt. I do this work because I love it, but it's nice to be appreciated.
Bill, thank you for spending the time with us today. I hope listeners have now a clearer sense of Alzheimer's disease from front to end stages. And thank you again for listening to the show, for supporting it by way of listening. Please tell me what I'm doing well, what I'm not doing well, what you want to hear more of, and share that all over the social media on my accounts. I am on there. I am mentally not particularly well adjusted because of my connection with social media, but you can find me there. Let me know. Bill, thank you once again.
Sure. My pleasure.
Take care, folks, and bye for now.